Hello everyone, welcome back. I know it's been quite a while since my Osmaka video, but I'm back with the first part of the Tarsier project that I've been sporadically talking about for the past 2 months. It took me a bit longer than expected, as per usual, as I am a chronic procrastinator, but it's here now. And I want to start with a question. A weird question maybe, but a relevant one. What do Tarsiers and toads have in common? Off the bat, they don't seem to share that much. I mean, there's some similarities in physical appearance. For example, you could say they both have big bulging eyes, and they both have long hind legs perfect for leaping. You could even go a little bit further and say they're both primarily insectivorous, and they both rely on elaborate calls and songs to facilitate mating. And at that point, you might start to realize that there's actually a surprising number of parallels for two so distantly related animals. And it turns out that on the island of Sulawesi, those parallels don't just end at morphology and behavior. The tarsiers and toads of Sulawesi, along with many other taxa on the island, share a complex and interlinked history, a shared pattern of biogeography that can tell us something about the island's deep past. I'm going to assume that if you clicked on this video, you already know what a tarsier is. But on the off chance you don't, I'll give a very brief rundown. Tarsiers are haplorine primates. They are more closely related to me and you and baboons and marmosets and all the other monkeys and apes than they are to the strepsirrhines like lemurs and galagos and lorises. Despite their relatively close relationship to us, they look absolutely absurd. They've got these giant bulging eyes, long springy frog legs, and massive grasping hands. They spend most of their time hopping from tree trunk to tree trunk under the cover of night looking for insects. They are the only completely carnivorous primates, and they tend to be solitary, though new research keeps coming out to challenge that assumption. It's this very Inurin-esque morphology and diet that allowed for my toad comparison at the start of this video. And don't worry, that wasn't just an offhand, corny joke. The toads will be important later, so keep them in the back of your mind. Unfortunately, if you want to have a deep dive into all the oddities of tarsier anatomy and behavior, of which there are many, you'll have to come back another time. That's not going to be this video, but it's something I will absolutely tackle eventually. For our purposes, the most important thing about them is where they live. Tarsiers are exclusive to island Southeast Asia. They live only in 3 major islands of the Malay archipelago, Sumatra, Borneo, and Sulawesi, and in the southern islands of the Philippines, notably Mindanao, Bohol, Leyte, and Samar. They are completely absent from mainland Asia. This distribution on its own is already notable. The Tarsiers of Sulawesi are one of the few vertebrate groups to span the famous biogeographical gap that is Wallace's Line. Wallace's Line lies between Sulawesi on the east and Bali, Java, and Borneo on the west. It marks the end of the traditional Southeast Asian faunal communities of tigers, rhinos, elephants, and other mammals. Sulawesi, the Moluccas Islands, and the Lesser Sunda Islands form their own biogeographic region known as Wallacea. This region is something of a transitional zone between Asian and Australian faunas, and the further you travel in the direction of either continent, the more of their respective faunas are represented. Wallacea is bounded on the east by the Lydekker Line, between New Guinea and islands like Halmahera, Ceram, and Timor. Keep these biogeographical lines in the back of your mind. They'll come up again later. Out of all of these islands, Sulawesi is the most important for us today. Sulawesi is an island within the Malay archipelago. It's the 4th largest island in the archipelago and the 11th largest island in the world. The whole of the Malay archipelago is located within the torrid zone, that's the tropics, and is blanketed in a deep, thick belt of rainforest. Sulawesi itself is one of the weirdest looking islands in the world. It's got a kind of flaily, tentacle-esque shape to it, almost like the letter K. It looks like the top arm is being pushed back by some sort of invisible, strong eastward ocean current. That northern arm is known as the Minahasa Peninsula. At its thinnest point, the Isthmus of Palu, the Minahasa is only about 12 miles across. Its northeastern tip is home to an arc of stratovolcanoes stretching out into the sea in the form of the Sangihe Islands. Jutting out to the east, we find the Eastern Peninsula, to the north of which lie the Togian Islands, and to the south of which lie the Palang Islands. To the southeast, we have the Southeast Peninsula, to the south of which lie the large islands of Wawoni, Buton, Muna, and Kabina. And then, traveling straight south from the central portion of the island, sits the, once again very creatively named, South Peninsula. The entire island is quite mountainous, with very little low-lying flatland. As such, it's divided into two major ecoregions, the montane rainforest in the highlands, and the lowland rainforest in the valleys. Sulawesi is big, but not massive. It's small enough and climatically and geographically homogeneous enough to support a similarly homogeneous ecology. And I'm going to leave our discussion of the island itself at that for a moment. Until 2010, all tarsiers were thrown into a single genus, genus Tarsius, within the monogeneric family Tarsidae. This genus usually was said to contain three species, Tarsius spectrum in Sulawesi, Tarsius bencanus in southern Sumatra and Borneo, and Tarsius serichta in the southern Philippines. Each had a few subspecies scattered about their range, often on outlying islands or in remote separated populations. There were a few morphological distinguishers between them, but by and large they were all considered to be very similar species. But as the late 90s and 2000s rolled around and genetic sequencing became more widely available, it was immediately clear that divergences between these lineages were much, much deeper than previously assumed. It seemed that these three species had all split from one another at least as far back as the Miocene, and potentially as long ago as the late Eocene. These massive genetic divergences were so surprising that they were used in 2010 by Colin Groves and Myron Shikella to elevate each of these three species into three separate genera. Tarsius was maintained for the Sulawesi populations, while the Sumatran and Bornean population was given the name Cephalopacus. The southern Philippine group was named Carlito, after the late Carlito Pizarras, an important figure in tarsier conservation in the Philippines. While the proposed taxonomic revisions were mainly focused on the genetics, there are real morphological differences between the three genera. For example, they differ in size. Cephalopacus is usually the largest, while Tarsius is on average the smallest. They differ in the amount of hair coverage on their tails and in how many sets of nipples they have. Cephalopacus has only two, while Carlito and Tarsius each have three. Furthermore, they differ not just in the actual nucleotide sequences of their DNA, but also in the actual physical structure of the genome itself. Tarsius has 46 chromosomes, just like us, while both Cephalopacus and Carlito have a whopping 80 chromosomes in each of their cells, the most of any primate. They also differ in sociality. Genus Tarsius is quite social, often living in family groups or as mated pairs, while Cephalopacus and Carlito are primarily solitary. Although again, as I hinted at earlier, there's new exciting research out of the Philippines that challenges this narrative. I won't get into it here, but there's growing evidence of sociality among Philippine tarsiers as well. Cladistically, the three genera map out like this. Tarsius is our earliest diverging lineage, and Cephalopacus and Carlito are sister taxa. The node separating genus Tarsius from the others has been recently dated to more or less 22 million years ago, right around the Oligocene-Miocene boundary. The node separating Cephalopacus and Carlito has been dated to around 10 million years ago in the late Miocene. This pattern in and of itself is interesting, for reasons we will eventually get into having to do with the region's paleogeography. The fact that the oldest lineage is isolated on Sulawesi is important, so keep that in mind. Within genus Cephalopacus, diversity is relatively low, right? We've got one species, Cephalopacus bencanus, with 4 recognized subspecies. You got Cephalopacus bencanus from Sumatra, Cephalopacus bencanus borneanus from Borneo, Cephalopacus bencanus saltator from the island of Belitung, and Cephalopacus bencanus natunensis from the Natuna Islands. But I should emphasize that these separations are rather understudied and may not wind up being valid when more research is conducted. The major lineages on Borneo and Sumatra are almost certainly distinct and likely diverged from each other around 7 million years ago during the late Miocene. But the two small island lineages are much younger, if they're even valid at all, potentially diverging within the last 20,000 years. Genus Carlito also only contains one species, Carlito serichta, which is traditionally separated out into 3 subspecies, Carlito serichta fraterculus from Bohol, Carlito serichta carbonarius from Mindanao, and Carlito serichta from Leyte and Samar. But more recent molecular work by Rafe M. Brown and colleagues has challenged this framework, identifying a completely different branching pattern than the subspecies classifications had previously called for. The Bohol and Samar Leyte population appear to be genetically one and the same, and a completely novel lineage centered on Dinagat Island and the Surigao Peninsula in northern Mindanao was discovered, called the Dinagat-Caraga group. The rest fall into a general population gradient across Mindanao, and potentially separating out into a Mindanao group and a Zamboanga group. Again, diversity here is fairly low, but all these lineages seem to be pretty young,
like at least within the last 3 million years young, if not within the last 20,000 years young. Brown's group doesn't give any estimated divergence dates, but again, given the paleogeography we will eventually discuss, anything outside of 3 million years wouldn't make all that much sense. My hunch is that these lineages are very young, like Holocene young, potentially. Same with the Bellytongue and Natuna Islands populations. And I'll explain why eventually. But for now, let's turn our attention to the star of the show, genus Tarsius. Genus Tarsius, which again is our outgroup with respect to Cephalopacus and Carlito, is remarkably diverse. While each of our other genera are monospecific, meaning they contain only one species, genus Tarsius contains at least 12 named species, and almost certainly even more that have yet to be described. It seems like every little peninsula, every little cove in Sulawesi has its own Tarsier species. Groves and Shikella named 9 of these species in their initial 2010 study, and 3 more have been subsequently described. In the far northeast of the Minahasa Peninsula, we have Tarsius spectrum gerskiae. Following the peninsula west, we find Tarsius supriatnii. Sandwiched between these two is a currently undescribed population that may represent a unique species, known as the Labanu form. Moving south from here, we have Tarsius wallacei, inhabiting the Isthmus of Palau. Heading into the mainland, we find ourselves face to face with Tarsius dentatus in the east and Tarsius lariang in the west. Throughout this region, up in the highland cloud forests above 6,000 feet, dwells Tarsius pumilus. Further south still, we find Tarsius fuscus and another hypothetical, as of yet undescribed species near the city of Kendari, on the southeast peninsula. On the very southernmost tip of the southern peninsula sits another likely distinct but still undescribed population called the Dessa Ara form. Out in the Palang Islands, Tarsius palangensis makes its home, while Tarsius nemetzi inhabits the Tokian Islands to the north. Far to the north, on the island of Sangir, lives Tarsius sangirensis, while further south in the same arc, Tarsius tumpara enjoys the beautiful views from the volcanic island of Siau. On the far opposite side of the island, Tarsius tarsier inhabits the island of Salayer. 2 more possibly distinct but as of yet undescribed species exist just south of the southeast peninsula, one sharing the islands of Muna and Bhutan, and another on the island of Cabena. Though I should note, some publications lump these 2 groups together. Lastly, one more, still hypothetical species may exist on the 3 islands of Bunikan National Park, though whether or not this population is awarded the species title remains to be seen. If we include all of the undescribed populations as distinct species, that brings our genus total up to 18. That's 18 species in Tarsius, as opposed to 1 in both of our other genera. Even a more conservative estimate hovers around 14 species. This leaves me with 2 questions. Firstly, why is genus Tarsius so diverse, right? Why is Sulawesi, a rather small island, at least compared to Borneo or Sumatra, home to at least 12 times the diversity of Tarsier taxa? And secondly, why did it take us so long to notice this? If Sulawesi tarsiers are this crazy mishmash of a dozen or more different species, why did it take us until 2010 and beyond to spot the differences?
I'm going to answer the second question first, and for that, I need to introduce the concept of a cryptic species. These are species that are morphologically identical, or at least almost identical to one another, but are greatly diverged genetically or behaviorally. In an anatomical examination, you can't tell them apart, but a deeper investigation of their genes can show you just how distinct they really are. A good example of this is the African elephant. For a long time, genus Loxodonta was thought to have one species, right? We had one species of African elephant. But it wasn't until 2001 that we split that up into 2 separate species, the African forest elephant and the African bush elephant. And that decision was almost entirely based on their deep genetic divergence. And while some morphological differences were noted after the fact, mostly having to do with size, it took the discovery of the genetic differences to allow us to spot the morphology. Alternatively, you could look at the Tapanuli orangutan, the 3rd species of orangutan that was hiding in plain sight in southern Sumatra for years. But it wasn't noticed until 2017 when researchers sequenced the population's DNA and discovered that the Tapanuli group was in fact the most basal extant orangutan lineage, well outside of the split between the Bornean and Sumatran populations. Turning away from vertebrates, we can look at lacewings, where what was initially thought to be one species, Chrysoprula carnea, was parsed out into a ton of different species to create the Chrysoprula carnea species complex. Visually, they all look identical, but the songs they buzz out to call mates are very distinct. This song discrepancy is what researchers initially noticed, prompting them to rethink the single species designation. Their suspicions were later confirmed by molecular analysis, breaking up Chrysoprula carnea into 15 distinct species on the basis of their songs. Then there's the alder flycatcher and the willow flycatcher, which were thought to be the same species until as recently as 1973. In terms of morphology and coloration, the two birds are identical, but they sing completely different songs, and therefore do not breed together. Or there's the Rana-Pipian species complex, where what was initially thought to be one species of leopard frog turned out to be at least 27 separate species spread across North America, distinguishable only by their calls and their DNA. I could go on and on with examples of this. There are countless different groups of creatures where the species lines are murky, if not mostly invisible. But I think you get the picture. Now, with those last three, you may be noticing a specific theme. While their morphology might be completely or practically identical, often their behavior, specifically their vocal behavior, is not. The study of the production and perception of sounds among organisms is called bioacoustics, and it has been used to identify many cryptic species like our aforementioned flycatchers and lacewings and frogs. Bioacoustics played a crucial role in breaking up Tarsier spectrum into what is now called the Tarsier species complex. You see, the Tarsiers of genus Tarsius, like the birds and frogs and lacewings, sing. They frequently live in small family groups, usually consisting of a pair-bonded male and female and their offspring. And every morning, as they return home to their sleeping sites from their nightly hunt, these male and female pairs will engage in a sort of duet. They'll often hit notes that are incredibly physically demanding for them, and it's been hypothesized that sexual selection has played a key role in this trait's proliferation, with the quality of the song essentially serving as a proxy for overall fitness. Basically, whoever has the most energy to spend on frivolous things like singing clearly must be healthy and fit, and is therefore an attractive mate. I think there's more to it than that. It probably also has something to do with maintaining the emotional bond between a monogamous pair, but sexual selection has certainly shaped it in many ways. This duet singing happens only in Sulawesi, and seems to be a Tarsius-specific trait. Neither of the other two genera appear to have anything analogous. The exact types of song being sung have been crucial in identifying the different species spread out across the island and its surrounding satellite islands. Generally speaking, a different song means a different species, and genetic testing confirms it. So, despite the overall nearly identical morphology present across the Tarsier complex, it's been possible to break it up into at least 12 and almost certainly more distinct species. Bioacoustics tells us there's at least 16 different song types throughout the island, meaning there are probably 16 total species, many of which have yet to be named and described. Molecular clock estimates place the divergences between most of these lineages fairly recently, with a major split between two roughly equally diverse clades happening about 2.5 million years ago. The clades appear to be split fairly evenly north to south, with the exception of the potential Kandari species, which groups with the northern species but is found in the southeastern peninsula. But there might be something else going in here, because that also appears to be the most basal northern lineage. I don't know. However, most of the actual speciation of these groups within these two clades, so the actual diversification of the species we see today, is happening between about 1.6 and 0.1 million years ago, and perhaps even more recently. Seeing these numbers, one might question the wisdom of insisting that two Tarsius populations, separated by less than 100,000 years, are full-fledged species, while simultaneously holding that the Bornean and Sumatran subspecies of Cephalopacus must fall under the same species umbrella despite having a divergence of almost 7 million years. But the vocalizations do deeply affect mate choice in Tarsius, so even two very recently diverged groups that have different vocalizations would not, under normal circumstances, recognize one another as mates, essentially forcing reproductive isolation via behavior. And no such behavior exists among Cephalopacus or Carlito, so perhaps the distinctions here are warranted. Regardless, there is one particular species within genus Tarsius that is of note. There's one member here that falls outside of the Tarsier complex. That's Tarsius pumilus, the pygmy tarsier. This is the smallest tarsier species of all, and it lives solely in the highland regions of Sulawesi. Interestingly enough, pygmy tarsiers have no duet system, and they're significantly less social than their lowland relatives. They're incredibly diverged from the rest of the genus. Tarsius pumilus makes up the most basal lineage, likely diverging about 9.9 million years ago, long before the speciations of our 2.5 million year old branches. So, what do we make of all this? This is extremely strange, right? We have one massive region in the form of Borneo and Sumatra and the southern Philippines that houses only two tarsier species and relatively low diversity of different lineages. And then we have one comparatively tiny region, that's Sulawesi, housing a massive diversity of distinct Tarsier lineages that seem quite resistant to gene flow. Yes, there's some gradation between populations in Sulawesi. There are hybrid zones, and there seems to be some introgression every now and again. But it's much more rare than it should be for populations that are so recently diverged and in literal physical contact with one another. So, what the hell is going on here? This is so bizarre. It's an absolutely ridiculous biogeographic setup. And this, right here, is the Tarsier biogeography problem. It doesn't seem to make any sense. How did this happen? Now, there is an answer. The problem has been solved. But I think I'm going to leave you with a little bit of a cliffhanger here for now, and pivot back to talking about the island of Sulawesi in a broader sense. The actual ecosystems throughout Sulawesi are very similar regardless of where you travel, assuming you stay either within that highland ecoregion or the lowland ecoregion. But there's something strange going on beneath the surface. This isn't just a Tarsier story. While the organisms you find anywhere on the island might look similar and act similar, the actual populations are deeply fractured. This is where those Sulawesian toads I mentioned at the very beginning come into play. Ingurophrynus is a genus of toads that are found throughout Southeast Asia and its surrounding islands. They're pretty generic toads. They lack teeth and they secrete a bufotoxin on their back to dissuade predators. There are 12 species, one of which, Ingurophrynus celebensis, is native to Sulawesi. Throughout the entirety of the island, these toads are fairly consistent. They inhabit all kinds of habitats and are fairly versatile creatures. A toad in southern Sulawesi is going to look more or less the same as a toad in northern Sulawesi. But the real differences are lurking in the genome. If you sequence the genomes of a bunch of different toads from different regions around Sulawesi, you'll start to notice a pattern. Each of these regions has its own distinct genotype. Each of these genotypes represent a distinct breeding population that doesn't mix almost at all with its neighbors, just like we saw with the Tarsiers. We don't see a genetic gradient traveling across the island or radiating out into different populations. Instead, we see more or less distinct boxes, populations that breed only within themselves and have very little gene flow between them. And more strangely still, the borders between these toad populations match up noticeably well with the Tarsier species boundaries. This is weird. Again, there aren't any immediately visible geographic features separating these toad populations, and there's nothing that would physically stop them from breeding with one another. They just don't. And it's not just the Tarsiers and toads. If you pull up a map of the distribution of Sulawesi macaque species, you'll see almost the same pattern. They each inhabit their own little ecological slice of the island. And the more you look, the more you start to see this pattern everywhere. The flying lizards of the genus Draco and the fanged frogs of genus Limnonectes also follow a similar distribution. Even large land mammals like the Anoa or the Sulawesi warty pig and the Beberusa seem to reflect this same pattern of diversity when you map out their population structure. I mean, it's just map after map after map. Shrews, mice, even cockroaches. All these populations seem to have the same, or at least vaguely similar boundaries. And this isn't just a coincidence. These geographic regions that seem to contain distinct populations or species have come to be called Sulawesi's areas of endemism, or AOEs. These regions, despite being almost entirely identical in how their ecosystems function, act as separate, established ecological blocks with very limited gene flow between them. Where these blocks meet, the species form limited hybrid zones, where they interbreed and swap genes. Depending on who you ask and what species you're looking at, there are either 7 or 8 AOEs on Sulawesi proper, with outlying islands often having their own endemic genotypic groups. And again, there are no particularly noticeable geographic barriers between these regions. That's not to say there aren't interesting geographic features at these borders, but these are mostly low-lying depressions, not something that would cause any particular issue for an organism trying to cross from one to another. And again, we slam up against the question of why. I can't emphasize enough that there are no geographic barriers between these zones. So why did this weird, consistent pattern of diversification occur across so many different, very distinct groups of animals? The answer to that question lies in the very rocks that constitute the island. But for that story, you'll have to wait for part 2, wherein our biological story will transform into a geological one, and we'll hopefully be able to parse out exactly how this situation came to be. But for now,
Thank you for watching. Have the best day. Go do something fun. Goodbye.